Hello, welcome to Bio 101 lab review and setup procedures for exercise 8, which was photosynthesis. So you'll grab your worksheet from the lab kit contents and you will see that lab 8, exercise 8 is photosynthesis, and the only thing you'll need from the lab kit is the 10 ml syringe. And it looks like this. It comes in a pack, all sealed. And then you'll look on the back and you'll see for exercise 8 photosynthesis, you need fresh spinach leaves, several. You'll need a 1/4 teaspoon of baking soda. You'll need 1 teaspoon, 1 hole punch or straw, and 1 table lamp or light source. And you'll see we have our table lamp already set up here. You can use something like this, or you can use a lamp that you have sitting on a table in the living room. Now, I have all the stuff from the kit and what I needed to provide from my house. And now I will begin with activity 1. Activity 1 is a virtual exercise, but we've written up the intro in your write-up, and it is observing photosynthesis via CO2 consumption. We remember from reading about photosynthesis that autotrophs use CO2 from the environment or its atmosphere along with water, usually as rain, and they produce their own foods, which is typically one form of a carbohydrate. Now, in the virtual lab, you will see a demonstration of a tiny green plant called Elodea, and you are going to determine whether or not this Elodea plant, even though submerged in water, can carry out photosynthesis. So they're going to show you a pH indicator called phenol red, and phenol red is all about letting you know whether you're looking at something that's an acid or a base by changing color. So one of the most consistent chemical reactions that occurs out in the world is that CO2 from the atmosphere mixes with water and it forms carbonic acid. Think of the oceans of the world and the atmosphere that's in contact with the ocean and the turbulence of the ocean and all the movement that goes on. The first several meters of the ocean is rather acidic. It has a pH that could possibly be 6 or even 5.7 because it is creating carbonic acid. But this is not a stable compound in that environment. The hydrogen ions will break off, leaving a bicarbonate ion. And notice the arrows on either side of those equations are going back and forth because the hydrogen ion and the bicarbonate will reform carbonic acid. Carbonic acid will fall apart into CO2 and water, and vice versa. It's a constant cascade of events here that continues throughout time as long as there's water in the ocean and CO2 in our atmosphere. Now this phenol red comes into play because when carbonic acid is made, an acid is certainly different from the bicarbonate ion and certainly has a pH different from CO2 and water. We know just plain old water, typically you think of the pH as 7. So if we add phenol red to the water, you will see that you get sort of a neutral red color. And down here you see when the pH goes low, the phenol red will indicate a yellow-orange color. The more acidic it gets, the brighter the yellow. The opposite is true in a higher pH. The color turns bright pink, and the higher the pH, the brighter the pink. Now, when you go back to our equation up here with carbonic acid and CO2 and water, when the CO2 concentrations increase in aqueous solutions, it causes an increase in the concentration of hydrogen ions, thus decreasing the pH value. So this occurs through the formation of this intermediary compound called carbonic acid. And so what we can do, because we know plants use CO2, if we add CO2 to the water and make carbonic acid, we can watch the color of this indicator change as the autotroph uses CO2. It will actually pull the CO2 out of the water as the intermediate goes back and forth into its different formations. And when it's in the form of CO2, the autotroph can use it for the Benson cycle or the carbon reactions, thus removing CO2 from the solution and returning the pH back up to the neutral pH. So with little or no CO2 in solution, the pH should be approximately 7.6. The phenol red will actually be red. And the relationship between dissolved CO2 and pH can be summarized as higher CO2 concentrations result in higher hydrogen ion concentrations and thus lower pH values. So it's an inverse relationship. The higher the CO2, the lower the pH. The higher the pH, the lower the CO2 level goes. So it's like a seesaw. Now, the first thing you're going to do to accomplish this procedure is go to Blackboard, click on the menu tab Labs, go to the folder that says Exercise 8, and when you open that folder up, there's going to be two items. One of them is the virtual lab from McGraw-Hill entitled Photosynthesis, Carbon Dioxide Uptake, and you're going to click on that link and it'll take you out to Connect. You're going to start this exercise, and as it begins, you read the introduction, and it will start as phase 1. You are to choose a hypothesis and a strategy. There's two different things that you have to do to get this animation going. So the first one, you're going to choose the hypothesis. When you choose the hypothesis and you submit it, and it is correct, you're going to write your hypothesis in this blank space on this document. You're going to type it when you open it up on your computer. Type your hypothesis. Then you're going to pick your strategy. Of course, it says choose your strategy. You'll pick the strategy. You don't have to write that down, and you'll understand that as you're reading the Connect assignment. However, before it begins and you've got your hypothesis, remember a good hypothesis should contain your independent and dependent variables. So you want to be able to identify your independent and dependent variables in this exercise. Then you're going to name some standardized variables that are going to exist in this virtual lab that you'll be able to identify. You need to identify at least 2 standardized variables. 2. Then begin phase 2, and you'll click the button and it'll take you on to phase 2. Now, as you perform the simulation, you are going to use this table. There is a table online, and it will fill the table for you at the end of the exercise, but you are also going to fill it out on this table in this document. You're going to put what the contents are in test tube 1, which is on the left in the animation. Test tube 2 is on the right. What's in it? You're going to put the initial color of those 2 tubes, and then after the simulation, you're going to put the final color of both those tubes, as well as whether or not you think photosynthesis occurs. And you're going to put yes or no or plus or minus, indicate somehow or another that you believe photosynthesis occurred, and then answer the questions. So this particular exercise 1 is all online, and you'll be able to complete that document using the virtual information. Exercise 2, activity 2, is going to take a little bit more of your time than that one, and you can't do it anywhere but at home. So this exercise is measuring photosynthesis by what we call a floating leaf assay. Now, there is a nice illustration here of a cross-section of a leaf. This is showing you the top of the leaf. Excuse me. This is the bottom of the leaf. They both have an epidermis. So there's an upper epidermis and a lower epidermis. In case you get confused, you can always look at the epidermis to see whether this is the top of the leaf, this is the bottom of the leaf. Excuse me. Now, there are the stoma on the bottom of the leaf that allows for the exchange of gases, as you read in chapter 8. But what we're going to do, since we know inside these cells, the mesophyll, there are chloroplasts. Chloroplasts are filled with the thylakoid membrane. The thylakoid membrane has the pigments, and this is where photosynthesis takes place. So what we're going to do is we're going to take advantage of, you see how loose these cells are and there's space in between them? Well, that allows the cells, the leaf and the tissues to fill and exchange CO2 and oxygen. So we're going to take advantage of this space that's here. And we're going to take out the air that's in there, CO2 or oxygen, and we're going to fill up that space with a liquid. And that's going to cause the cells to, the leaf tissue, to fall to the bottom of a beaker filled with water. So what then you think, well, if these leaves are on the bottom of a beaker, they won't have access to CO2. And CO2, they need the carbon from the CO2 in order to carry out the carbon reactions or the Benson cycle or the Calvin-Benson cycle. So what we're going to do is replace it with sodium bicarbonate solution. So you've got these big, large air spaces in the tissue. The air spaces are much less dense than much anything else, and leaves will float on top of the water. But if we replaced the air spaces with solution, doesn't matter whether it's straight water or sodium bicarbonate solution, the overall density of the leaf will increase, and the overall density of the leaf will have the leaves infiltrated with sodium bicarbonate, and it increases and the disc will sink. And if we have one of these solutions filled with sodium bicarbonate, if you look at the molecular formula, it is NaHCO3. There is carbon in that solution. So plants are not picky. If they're not exposed to the atmosphere of CO2, they will use the carbon from wherever they can find it. And sodium bicarbonate has carbon. They are going to use the carbon from the sodium bicarbonate solution in order to carry out photosynthesis. If they're carrying out photosynthesis, they're going to produce oxygen. And when the gas is made, it will fill up the space where the water is, or the solution is, and it will force it out of the stoma, thus filling this space with air or oxygen again, and that will cause the leaves to rise to the top of the water. So it's going to change the buoyancy if the leaves are able to carry out photosynthesis. That's the whole idea behind this exercise. So you have what you need to pull, the procedure here, what you need to provide from the student. Here's what we provided, your beakers, your syringe, and the disposable pipettes. So From there, you're going to follow the steps indicated, and there's also a picture here that you can refer to, and I am going to show you how you can proceed. So you have your two beakers. One of the first things you want to do is, these are 250 ml beakers. So I put in approximately 150 mils of water. They are labeled. Each beaker is labeled. That one's water. This one is going to be sodium bicarbonate. So the sodium bicarbonate is labeled and the water is labeled. Why in the world would we be using a beaker with just water? Hopefully you'll know the answer to that. So now the instructions tell you to do is take a pinch or an eighth of a teaspoon of the sodium bicarbonate. And this is baking soda. This is the baking soda that comes right out of your kitchen in the yellow box, Arm & Hammer. And I'm simply going to take a pinch. Okay, I'm not going to use an eighth of a teaspoon. I'm going to take a pinch and I'm going to sprinkle it in my water, just like that. And that's all you need is the pinch. So once I have the pinch of sodium bicarbonate, I am going to take my spoon and I am going to stir this up and make sure it will dissolve. And it's just a small little amount. I'll leave my spoon in there so I know that spoon was the spoon that I used for the sodium bicarbonate. Now, after you do that, you're going to take your soap. If you have Dawn or Palmolive or whatever, Arm & Hammer, whatever kind of soap you have, we've got just a little bottle of soap. I'm going to put a drop or two in each beaker. The soap is simply a surfactant that will prevent hydrogen bonding between the glass and the leaves. I'm going to just use a pipette for this one to keep it stirred up. Now you're not stirring it up trying to make bubbles or anything like that. You're just allowing the soap to get in there and prevent hydrogen bonding from forming. Now, you can either use a straw or a hole punch. I have a hole punch. I just got a regular old hole punch. Here are my spinach leaves. They're nice and firm. I pulled a couple of them. Now I will say that if you can, I would put down a piece of paper, a paper towel, something so that these little hole punches don't stick to the table, because they will, and it makes it hard to get them off the table without crinkling them up. So I am simply going to punch a hole in my spinach leaf. And the instructions tell you that you're going to need 25 hole punches. So you can see that it simply makes really nice, clean hole punches. The firmer the leaf you got, the easier it is. As you can see, I can go much faster with this one because the leaf is a little firmer. So you are going to punch out 25 of these, and you can do 10 at a time if you'd like. You just have to evacuate them a lot, several more times than doing it all at once. So you can see I've got a lot of hole punches out of my leaf, and they're here in my hole punch. This one happens to have a little guard on it, so it collects them. And so there is my pile of hole punches. And like I said, you want to try to keep them either on the paper or in your hand, just so they don't stick, because when they stick to something, then you have to curl them up, and that tends to make them not work quite as easy. So here are my hole punches. We call them chads or hole punches, but I've got bunches here on my paper. Now, you are going to take your syringe, right? And this syringe, take the plunger out, and I'm going to take my hole punches, and I'm trying to avoid touching them as much as I can, and I am going to pour the chads, the hole punches, into the syringe. And because they want to stick to your hands, it's easier to use the paper. And as you see, they all didn't necessarily go in. They fell off to the side, and it's okay. I'm just picking them up and I'm putting them back in my syringe. So I have, as you see, quite a few hole punches or chads in my syringe. I'm going to put my plunger back in my syringe. I am going to blow the air out of my syringe, go down about that far. You don't want to crush your chads because it will damage them, and then you'll have to do more. You'll have to cut some more. Now I'm going to do my sodium bicarbonate solution first. So I'm going to put, and I'll swirl it around again, I'm going to put my syringe in there, and I am going to draw up solution, that sodium bicarbonate solution, into my syringe. And you see here are the little hole punches from the leaves, the spinach leaves. I am going to put my thumb over the top, and I'm going to pull down on my plunger, and you'll start to see bubbles. Bubbles will start forming, and you will hear a vacuum formed. You can hear that. Now I'm going to push up, and I'm going to push all that air out. Don't crush your chads. Pull another vacuum. Keep them in the solution if you can. So I just wiggle and shake. You see all the bubbles. Let the vacuum off. Push the air back out. And I do this about 3 times. You see now they start falling. By the 3rd time, I have pulled all the air out of the mesophyll, and it has been replaced with the sodium bicarbonate solution. So these guys now will sink. You see they're filled with solution, and they will sink to the bottom of your beaker. So I come over here to my beaker. I am going to, see if I can get this close enough for you to see. I am going to open my syringe over my beaker, and the majority of them fell into the beaker. Well, all of mine did. But if you have some that are stuck, put your plunger back in there, try to scoot them down to the bottom, go back into your beaker, draw up some more solution, shake it around, and get your little hole punches loose. Then hold it over the beaker and just pour them out into the beaker again. Now you want to look at this and ensure, I think I'll do this where you can see it better. You want to be sure that your beaker has all these chads go to the bottom. And I can easily see that I've got at least 15 on the bottom. And so now what I'm going to do is I'm going to turn on my table lamp. This guy goes underneath the lamp right there. Now I'm going to watch that, and as photosynthesis takes place, and if it does, if there's carbon there, then photosynthesis will start filling up these things with oxygen, and they will start rising to the surface of the water. And that's what you want to do. This one is plain water, and you're going to take another leaf. You're going to cut out 25 more chads and do the exact same thing. Rinse out your syringe that had sodium bicarbonate in it with water. Then draw up, put your chads in here, draw up water from this beaker and empty the chads into the beaker with just plain water. And then you are to observe this and fill in your table. So we've got a table here that you're starting your timer. The minute I put that under the lamp, I started the timer. 1 minute. After 1 minute, I'm going to look at my beaker and see how many discs are floating in the bicarbonate solution. If there's 0, I'll write 0. Then after 2 minutes, there may be 1 or 2, and I'll record that. And I'm going to go all the way down here for 20 minutes and see if I can get all the chads to come up to the surface. If all of your chads come up to the surface and say it's only been 12 minutes, well, you can stop the experiment for that beaker because all of your discs have already floated to the top. And you can say with confidence that your data supports your hypothesis. And then you're going to do the same thing with the water. All right, that would get you through this activity, the second activity with photosynthesis. And you will need to watch your beakers in order to record your data. If you have any questions, let us know, and we'll see you during exercise 9.